Thank you for the kind introduction. And so today I'm going to give a little different talk than the last one. This is going to be focused really on how we're trying to use direct RNA sequencing to start to disentangle the neural mechanisms of substance use disorders. And just to try to quantify the problem a little bit, over the most recent set of data of a few years from 2019 to 2022, there's almost a 400% increase in the population of people who opioid overdosed that also had concomitant xylazine on board at the time in clinical and postmortem clinical testing. And this isn't just an American problem. This has started to become a global problem because back in May of 22 was the first reported cases of xylazine on board during opioid overdose here in London. Now, originally, it was very hard to model this preclinically in an animal model system. You can get animals to self-administer opioids, you can get them to self-administer cocaine or other addictive substances, but xylazine is naturally aversive. In case anyone's not familiar, xylazine is an alpha-2 adrenergic agonist, and so the sedative effects of the drug itself make it so that the animal doesn't want to continue to push the button in a self-administration context and actually continue taking the drug. However, with Cassie Gibson's group at the University of Kentucky, they were able to do this really interesting behavioral training phenotype where they're actually able to pre-train animals on fentanyl self-administration, and then we're able to switch them over to self-administering fentanyl plus xylazine, and so they'll take this, this actually tagged fentanyl now. And when we do this, the animals will self-administer, sort of like we would expect for the fentanyl alone, that they'll escalate dose over time, and that they'll maintain this out to 10 days of self-administration, which is fairly standard in the substance abuse fields. Now, we took this and we used the brain tissue from the nucleus accumbens. It's a region specific to the mesolimbic pathway and substance abuse. It's associated with volitional taking and the experience of reward. We use that RNA for input to the RNA-004 flow cells and the related kits, and I have the analysis platforms there we use as well. Now, when we looked at the data we got out of this, and we looked in the context of fentanyl or fentanyl plus xylazine compared to the animals that just were able to self-administer saline in the later training times, we see differences in gene expression. When we compare these gene expressions, because this is a bulk RNA-seq experiment here, to prior published literature from our group in single cell from the same brain region, we see a lot of overlap at the gene differential expression level. Where this got a lot more interesting for us then is when we start to look at the isoform differences. And so we see nearly twice as many differentially expressed isoforms in this data as we do from the genes themselves. And we see sort of all combinations that one might expect here. We see genes that are driven by differential expression of single isoforms, sometimes multiple isoforms, or interestingly, a handful of genes where the differential expression is driven by no specific isoform, but just maybe a general upregulation and tone of the gene itself, so across multiple. And we also see some interesting examples, which I don't have all the pileups for time reasons, but you actually see examples where there are two different isoforms, one up, one down, so isoform switching, that you can't actually appreciate at all at the gene level. Now, interestingly, in this xylazine adulterated fentanyl model of preclinical self-administration, what we don't see, surprisingly, is much overlap. And so if you just look at the Venn diagrams at the bottom there, the genes and the isoforms that are differentially expressed are almost 80% independent between the two conditions, which is really radically different than anything we would have expected going into this. And so this really helps quantify the scope of the problem that we're dealing with and understanding why xylazine causes so many problems and increases the relationship to opioid overdose. There's actually preclinical literature and clinical case reports now available that shows that individuals who have xylazine adulterated fentanyl in their system are actually resistant to opioid overdose treatments. And so now you're having something that makes the sustained high last longer in the abusers, but now makes them treatment resistant. Now, to try to also understand how this was changing the epigenetic component of this, I'm showing methyl-5-cytosine and methyl-6-adenosine in the DROC context. And so we can find epitranscriptomic modification across the entire genome. It's not specific to any one thing. It's not specific to, and this is site-specific. This is single base here in the pileup. And this isn't specific to just the genes that are, or isoforms that are differentially expressed. We actually find it throughout the entire genome. If you try to express this to make it a little easier to see as the absolute difference in quantification, we then start to see a lot of differences because now all of a sudden you have a 100 to even sometimes 500 and 600% change in the amount of differential methylation that we see at some of these individual base pairs. When a lot of these start to line up then, just to give a few examples for the group here then, to try to give some specifics, you can also, because you're doing this in a bulk context, you can start to appreciate the mitochondrial genome as well. And so we see some individual differences on those genes for the methylation differences. But in the modified bases, we also find genes that we know have we know have roles in opioid addiction. And I'm showing RGS9 here because that's one of particular interest to my lab. The gene RGS9 is associated with differences in opioid dependence, specifically at the level of tolerance. So SNPs and RGS9 are associated with difference in the continued volitional self-administration of opioids. And in this data, we can actually identify differences in epitranscriptomic modification. And so a question we're going to want to dig into here in our upcoming work is one to validate these all independently then by other methods, but then also see what's the translational differences that these lead to in these specific genes and other genes we know are associated with opioid self-administration. And with that, I'd like to thank the members of my lab and the members of Cassie Gibson's team at University of Kentucky who helped us generate the self-administration data, and I'd be happy to answer questions.